questions, and focus on atherosclerosis just because that's the theme of the seminar series. So as Ebru said, I'm at the University of Tennessee Health Science Center, which is in Memphis, Tennessee. And I have little disclosures where I'm on the scientific advisory board of Macrophage Pharma, and my husband has a company, but neither of these have to do with the talk today. We know that obesity, diabetes, and other comorbidities are increasing and major problems in the US. And a couple of years ago, we published a review on adipose tissue and cancer, but actually a lot of the review is just looking at the immune system and adiposity. And so my student and I, and mostly my student, spent a lot of time looking at changes in adiposity over time and looking at all the dynamic changes to this incredible tissue, which we see changes in extracellular matrix, immune cell infiltration, proliferation, changes in phenotype, cell death, etc. And so I'm just going to briefly show you that some cells can rapidly infiltrate and then kind of leave with time. Some cells are more present in the lean state, and with increasing obesity over time, you see decreases in like Tregs or eosinophils. And these changes generally, or I should state that Tregs and eosinophils are generally thought to be more protective, like in the lean state. And then lots of cells will increase with obesity over time that are typically thought to be negative players in insulin resistance. And then, of course, my favorite is the macrophage, which is present at high levels and then can increase. And I'm going to spend time today talking about macrophages. And a lot of these changes we see so dynamically in adipose tissue are also happening in blood vessels, liver, etc. And so we're interested, and this series is interested in cardiometabolism, and we know that heart disease is a problem. And I, although Ebru and I met in Boston, I spent 5 years as a postdoc in Duke, and then 8 years as faculty at UNC Chapel Hill. And my husband and I were recruited to Memphis, to the University of Tennessee Health Science Center. And you see in the logo, this is the shape of Tennessee, which is right here. And we are located right at the teeny corner of Tennessee. And if you look, that is the deepest red. Pretty much any image you're going to look at, you know, COVID cases, obesity, diabetes, heart disease, poverty. We are in the epicenter of a place that really needs help. So that will be my first call, is that anyone that's interested in coming to speak here or travel here or collaborate, and when the virus is over, we'd love to host you. But so today I'm going to look and talk about what I'm really interested in, which is how cells move and talk to other cells, and where things go. So I love to think about where substrates go inside of a cell. And we know that macrophages are really dynamically changing with metabolites such as glucose or fatty acids, or if they engulf dead cells like adipocytes. And so the subtype and phenotype of a macrophage is really dependent on the microenvironment, cell-to-cell communication, and the nutrient milieu. And so I'll just review some literature that pretty much comes from like mid-2000s and has exploded, as you all know, with macrophage metabolism. So some recent work showed that we can have a resting macrophage, but actually several macrophages, mostly resident macrophages in tissue, are derived from the yolk sac or the fetal liver. And these primarily make up tissue resident macrophages. And they are proliferative, which is really interesting because you normally think of a macrophage as a terminally differentiated cell. So cells can, of course, macrophages can infiltrate tissue from a monocyte, so a monocyte-derived macrophage. And classically in vitro, there's lots of work showing that if you stimulate macrophages with certain stimuli, they have a phenotype. So if we stimulate with T helper 1 type cytokines or lipopolysaccharide, you can make this classically activated M1-like macrophage. And these are typically associated with metabolic dysfunction, like atherosclerosis or obesity, diabetes. Whereas in contrast, you can have this M2-like alternatively activated macrophage, which is stimulated by Th2 type cytokines or allergic response. They're very involved in homeostasis and tissue maintenance, extracellular matrix remodeling. But these are broad strokes. These are in vitro models. The reality and what's been shown over the past decade and more is that there's this huge difference and phenotype differences depending on which stimuli that the lab is interested in, and people can make up different names. And we know iron is important from Alyssa Hasty's work. So the take-home message is, of course, that macrophages are very complex and dynamic with a mixed phenotype. What my lab is focused on is the role of metabolism. So it was well known, and now increasingly well known, that the pro-inflammatory type macrophage is dependent on glycolysis, whereas the alternatively activated macrophage can be more dependent, mostly dependent on oxidative metabolism, mitochondrial oxidation, like fatty acid oxidation. But in reality, it's more like a flux, and we have this overlapping phenotype, and that's what I'm going to talk about today. So we have our cell and our little mitochondria. And like I said, I'm really interested in thinking where things come from and where they go, and whether that's a cell or a metabolite or whatever. My lab is mostly focused on glucose and fatty acid metabolism, because amino acids are just too intimidating for me. And what we do is use transporters like a door to open and close the door to a certain substrate. So we've looked at glucose transporter, GLUT1. We've looked at fatty acid transporter, FATP1. And so today I'm going to talk about our GLUT1 project, which we published some early work in JBC and recent work in Journal of Immunology. But I won't talk about my studies on FATP1, which is a very long chain fatty acid transporter on macrophages. But that was published, obesity work in molecular metabolism, and our atherosclerosis work in 2017 in the journal Atherosclerosis. So glucose is transported by GLUT transporters, and there can be 12 glucose and other hexose transporters like fructose. It's a transmembrane protein, and we showed in certain macrophage subtypes, like most of the work I'm going to show you today is a bone marrow derived macrophage. And we typically use a really standardized way to study our cells in vitro. We leave them unstimulated, LPS interferon gamma, which is the M1 type, and then IL-4, which is the M2 type. And so we know that GLUT1 is really highly upregulated in murine cells as well as human cells, in the cells that are highly glycolytic, the M1-like stimulated cells. And so to kind of ask the question and model glucose metabolism, we first did a really simple study where we took a cell line, which is the raw 264.7 macrophage cell line. It's already actually kind of skewed towards an M1-like phenotype, but we overexpressed the GLUT1, and we were able to show that this can drive inflammation. So really briefly, I'll remind you that when you take an unstimulated macrophage and stimulate it with whatever stimuli, it becomes glycolytic. And when we drove GLUT1 expression, we were able to drive glucose uptake, metabolism, changes in metabolites, transcriptomics, and the end product was looking at some cytokines and chemokines, and we saw that these were upregulated, and this occurred even in the absence of any stimuli. So these cells, in the absence of stimuli, were really skewing pro-inflammatory. And we were able to inhibit this by inhibiting glycolysis with 2-deoxyglucose or oxidative stress, because we know glucose is very important in the pentose phosphate pathway. And then looking in vivo, we can stain for GLUT1 and see that it co-localizes to the crown-like structures, which are macrophages and other immune cells in adipose tissue. And then if we take out those adipose tissue macrophages and stratify by low or high GLUT1 expressors, we saw that the ones that are more GLUT1 expressing had higher amounts of cytokines. And this was done in collaboration with Jeff Rathmel. So, you know, we had this in vitro model, but we wanted to say, what would happen? Can we test the dependence of this pro-inflammatory macrophage on GLUT1? And we hypothesized that if we knock down GLUT1, if we knock down glycolysis, what's going to happen to inflammation? Are we going to decrease inflammation and skew it towards the M2 macrophage and show that this phenotype is dependent on GLUT1? So we were able to get the floxed mouse from Dale Abel, and crossing that with a myeloid specific Cree, the lysem Cree, we created a model, and we showed if you isolate out bone marrow derived macrophages, in the wild types have tons of GLUT1 and it's knocked out at the RNA level, it's knocked out at the protein level. We did metabolic studies, so we did tritiated glucose, and we see decreased uptake in BMDMs as well as peritoneal macrophages. We used C14 labeled glucose, and we could capture that C14 carbon dioxide to measure oxidative metabolism of this substrate, and saw reductions in oxidative metabolism of glucose 2 across the board. And so we further did some more bioenergetics to see exactly how responsive the cells are to glucose and other parameters using the Seahorse. And again, we have unstimulated. Red is the stimulated like M1, and blue is the IL-4 stimulated like M2. And you could see the cells are chugging along, but when we add the glucose, wild type cells take up lots of glucose, and when we uncouple with oligo, these cells can really take up and do a lot of glycolysis. But the knockouts have a poor response to glucose and a poor response to uncoupling in the LPS interferon gamma stimulated M1 cells. And so we've shown that we dramatically reduce glycolysis, and it's true for glycolysis, the glycolytic capacity, and the glycolytic reserve. So there's really no compensation, even though we know that cells express a little bit of GLUT3, and there's other GLUT transporters, but there's really no glucose uptake. We've also really recently shown that we're working with different inhibitors to GLUT1, and we can recapitulate the GLUT knockout phenotype. And so we wanted to ask, what are these cells living on? They're surviving in vitro. You know, we could find them in tissue. So we turned to metabolomics with Metabolon, and then some fluxomics with Jason Locasali at Duke. And all the data is in the paper. It's really kind of a data intense paper. I'm only going to highlight a few things today. So if we look at glycolysis and look at these intermediates, we know that glucose is not getting taken up into the macrophages. But in the GLUT1 knockouts, which are always going to be red, we see a blunting of all these intermediates of glycolysis, even when we go from unstimulated to the M1-like phenotype, where the wild types are using lots of glucose. The same for the end product lactate, much lower levels of lactate. And just representative here, but lots of the different metabolites in pentose phosphate pathway are downregulated. So we've really shut down this arm of metabolism. But so then what are the cells surviving on? What kind of other substrates or anaplerotic substrates might be involved? And we were able to use heavy isotope flux tracing of glutamine and see that we see increased glutamine flux through the TCA cycle, showing that they are reliant on at least other amino acids like glutamine, but then also due to metabolomics and some other changes in our fluxomics, we believe that there's an increased reliance on branched chain amino acids. And then lastly, I actually spent most of my graduate career studying fatty acid transport. So we were looking also at fatty acid metabolism in the absence of GLUT1, and using just fluorescent labeled, we see increases in fatty acid uptake, as well as, again, using a C14 labeled fatty acid, we see an increased reliance on a different substrate here. So more fatty acid oxidation in the knockouts. And again, we know that the M1-like macrophages are highly glycolytic. And in the cells that are highly oxidative, we see even in the knockout, lots of fatty acid metabolism. And so then last, we looked at transcriptomics to get some more insight. And as we suspected, because of the really downregulation of the pentose phosphate pathway, we see downregulation of some ROS pathways, and then downregulation of some pro-inflammatory signaling. And we confirmed like nitric oxide synthase was downregulated in our knockouts. And using flow, we saw less ROS biomarkers. And then I integrated gene expression transcriptomics with metabolomics using this website Bioanalyze, which it's really nice and it's pretty user friendly. And so, you know, as we expected, there was a lot less glycolysis. We saw more protective antioxidants, like the more glutathione pathways upregulated, decreased markers of oxidative stress, and I'm briefly going to talk about arginine and proline next. And so we know arginine metabolism, iNOS, arginase, is a huge marker in defining our macrophage phenotype. And this is a little cartoon from Metabolon, so it's a little bit ugly. But basically, we have our arginine, which is metabolized by iNOS into nitric oxide, you know, our RNS, our pro-inflammatory type of mediator, which also creates citrulline. In our knockouts, we see this downregulated of citrulline in GLUT1 knockout BMDMs. And then if we unmask the rest of the pathway, we actually see that the cells are much more M-like. So that arginase can metabolize arginine. So arginine either goes to iNOS or can go through arginase to make ornithine. And that ornithine is used for polyamine production or can be used for extracellular matrix remodeling. Both of these are very important in the M2 macrophage. And so we see, if we look at the metabolites for Metabolon, ornithine, much higher in our knockouts, and then we go down the pathway of polyamines, all of these are super high upregulated along this pathway in GLUT1 knockout, as well as these intermediates in collagen production, hydroxyproline. So this really suggests metabolically, this skewing. So then we thought, oh, this is such a home run. We've really reprogrammed these cells. They're going to be totally M2. And we saw some evidence of downregulation at the RNA level of some things, but then like MCP1, this chemokine was downregulated in our knockouts, but then other things were like either not regulated at the protein level or like wildly regulated like IL-1 beta. And this is really interesting because Luke O'Neill has published how important glycolysis is in the production of IL-1 beta and processing to produce the IL-1 beta protein. We actually don't see the lack of IL-1 beta. In fact, we see lots of variation and even in some cases more IL-1 beta. So if anybody has ideas on that, I'd love to hear. And so despite these pretty strong in vitro metabolic changes and rewiring of the substrate metabolism, we're not seeing this consistent change in the M1 or M2 inflammatory mediators. So to summarize the in vitro work, we did a lot of different approaches to show basically that we've successfully reprogrammed the macrophages. Glucose utilization is gone, the cells are healthy, they stick, you know, et cetera, and they're depending on other substrates. And this just shows how flexible macrophages can be. We've definitely showed reduced ROS production, and there's many different explanations like lower pentose phosphate pathway, but also some metabolites that we know are involved in ROS production, like itaconate, were increased in our GLUT1 knockout. And then last, it looks like there's an immunophenotyping more towards the M2 macrophage, but again, it's mixed. But M2 macrophages have high amounts of polyamines. We see reduction of some major pathways in pro-inflammatory. And with this mixed phenotype, I wonder if possibly we're looking at a stunted or premature phenotype. And those are things we're looking at now. I have a really great student in my lab, and I think he's on this, who knows a lot about MDSCs and myeloid precursors. And so getting back to atherosclerosis, that's our main question. So we wanted to take this model and ask what's happening with atherosclerosis, because it's kind of a complex and interesting phenotype in vitro. So we took this model that we've already generated where Cre crossed to the flocks. We took the marrow, we transplanted that marrow into LDL receptor knockout mice. These are irradiated, of course. And so the whole rest of the body is wild type for GLUT1 and can process GLUT1 and glucose fine. But in myeloid cells, and especially macrophages, which we know are so important to the atherosclerotic lesion, we have a knockout of GLUT1. And then we fed them Western diet and looked at what happened. So, you know, our goal and our premise was maybe if there's less M2 and kind of more less M1 and more M2-like, are we going to alter or protect the mice from lesion formation? And so first, to look at some of the basics, like the physiology. No differences in weight gain on these mice. And if you remember, we did a bone marrow transplant. We started them on Western diet, so they started gaining a lot of weight. No differences, and we also measured some blood pressure. And so some of the main parameters that might influence atherosclerosis, like glucose, HDL, triglycerides, were not different. No difference in blood pressure. However, we looked at plasma total cholesterol and LDL cholesterol. We saw significant elevations in our GLUT1 knockout transplanted mouse, suggesting that maybe the lesions are going to be bigger, of course. So then here we go. We looked at the aortic root and quantified with oil red O lesion size. No difference in lesion size in our wild type and knockout. We said, oh, the macrophages aren't getting there, of course. So we stayed for macrophages, no difference in macrophages. And I'm not showing the data, but we actually don't see any differences in macrophage infiltration to adipose tissue. In fact, we see more macrophages in adipose. And so it looks like there's not a defect in the macrophages getting to where they need to be. But I really, this part of the project was led by Liang Zhao, who's an amazing grad student. And she was like, you know what? I think the knockouts have this necrosis thing happening. And she really did incredible staining and histology. So you can see this is Masson's trichome stain, and you could see this necrosis. And so she quantified wild type compared to knockouts. So more than double of the mice had necrotic cores in our knockout transplants. So we're able to do digital histology using the Aperio, and we were able to quantify the intensity of the blue and the amount of the blue. And we saw overall that there was less collagen staining. And this is important because even though we saw no differences in the lesion sizes, for example, what's important to a lesion is the vulnerability. So an unstable lesion has a big necrotic core and a thin fibrous cap. And those are really fragile and are prone to thrombus. And that is what's going to kill the person with atherosclerosis. Whereas a stable cap is thick with collagen and is kind of more healthy, you can say, with less necrosis. So what we're seeing in knockout is really unstable lesion. And I'm almost done. So it was weird because in vitro was so dramatic. In vivo, kind of subtle, but there's a defect. What's going on, right? We see this necrosis. So Liang found that glucose can regulate actin. And I won't go into this, but there's some other enzymes involved in glycolysis that actually regulate actin and movement. And luckily at UNC, this was mostly done at UNC, there's a guy who studies cell migration and actin polymerization. So we worked with him to do a bead uptake assay. And you can stain them in different ways. So we had wild type and knockout. And so the beads can either stick to the outside of the macrophage, or if you permeabilize, you could measure beads on the outside and the inside. And so you could quantify adherent cells. So in wild type and knockout, we saw no difference in cells sticking to the outside. but we saw this significant difference in engulfment. So there's something going on where these macrophages can bind, but not internalize these beads. But beads, they're not LDL particles, they're not dead cells, it's just like a proxy measure. It engages different pathways. If you talk to somebody who studies phagocytosis, they're going to say that's like a really weak study, but we were excited. So then simultaneously, in a completely different way, Jeff Rathmel, my collaborator who was at Duke and now at Vanderbilt, was contacted by his friend and collaborator, Koti Ravichandran at UVA. So using bioinformatics and studying efferocytosis, which is uptake of dead cells, he came to the conclusion that GLUT1 is really important. So we were able to collaborate and show, again, in the knockout atherosclerosis model compared to the wild type, there's a lot more necrosis in the knockout. And he showed in lots of other ways, even zebrafish, the importance of GLUT1, SLC2A1, in taking up dead cells as being GLUT1 and glycolysis dependent. So to summarize, we show that GLUT1 is really a gatekeeper to this robust glycolytic response in macrophages, but we know that macrophages are highly flexible in lots of different substrate utilization. So these cells can survive and thrive without GLUT1. But our atherosclerosis studies show that there's some defects, and what we think is happening is that GLUT1, glycolysis dependent phagocytosis and efferocytosis was affected. And I want to acknowledge, so most of the work was done by my lab in North Carolina with Alex Freeman leading the in vitro and obesity work I didn't talk about today, and Liang Zhao leading the work on atherosclerosis. She's now a postdoc in New York City at Rockefeller. And then my lab in North Carolina, Ajith Pingley and Bin Tang followed up on all the Seahorse in vitro work. So I want to end by saying, this is where my lab is located, and everything I talked about today is actually not what I'm currently studying. So I want to end by saying that I hopefully in 1 week will be announcing that I'm looking for a postdoc in the role of obesity, microbiome, and the immune system in breast cancer. And as I mentioned, we study adipocyte and immune cells, which is really important as the soil where your breast cancer will grow. And so my lab is focused a lot, especially the past 3 years, on immunotherapy in triple negative breast cancer. So I want to thank my collaborators on the GLUT1 project and point out that Memphis is really fun, and literally right down the road is where Elvis recorded with Johnny Cash and all these great people. I want to thank my funders, and I guess I will take questions.
Thank you, Liza.
Sure.
I was hoping you would end with a Johnny Cash. Okay, short of the song. It was a great talk. Thank you so much for joining. So we'll get questions from the participants window. You guys can raise your hands as always. Should I stop sharing my screen, Ibra? Yeah, that's a good idea.
Okay.
And so while people are thinking about their questions, I have a question.
Yeah.
So Liza, it was really good that you broke down the paper. It was really heavy on metabolomics, and some people are not working on that so much. So I have a question. The macrophages kind of switching from glucose to using fatty acids and glutamine. I mean, they remind me of liver cells a little bit and cardiomyocytes. So which do you think they more prefer? Like, are they more like cardiomyocytes, burning fatty acids, or are they more like liver burning amino acids, or they can flex between?
Okay, that is a great question. And if anybody here studies T cells, T cells also are the same way. And I think that it really depends on the environment you're in. And it depends on for adiposity, it depends on the stage of adiposity. I think when you have an infection, an acute response, you need a lot of glycolysis to make your effector T cells, to have a strong antigen presentation by your dendritic cells, and to have a strong macrophage response. But in other cases, like this efferocytosis, dead cells, you do need the glycolysis to regulate. You know, immunity is always like hot and cold, so you need the Goldilocks happy in the middle. And I think metabolism is super flexible to regulate that.
Yeah, so that's really cool. We're learning about all of this now. There's so much to do. So I have a familiar name, Gökhan Hutamışlıgil, asking you a question. So Gökhan, go ahead.
Hi, Liza.
Hey, Gökhan.
Great work. Congratulations.
Thank you.
So the question I have is, if there is a strong role in efferocytosis, in the metabolic flux experiments that you do in culture, would that be, do you think, a potential confounder, or could that actually change the metabolic state of the cell? Or can you, for example, do those experiments in wild type and knockout cells by offering them some dead cells as meal, and then measure their fluxes?
Yes. So of course, we bought like fluorescently labeled LDL particles, and we were going to do dead cell uptake, but actually the reviewers didn't ask. And so we didn't do those experiments at that time. But of course, those are important in vitro experiments. But one thing that we've optimized in our lab, and we're doing this in tumors, but you can do in any tissue. And I should say Mehdi Chaib, my graduate student, did this completely himself, is that you can label epithelial cells or whatever with whatever stain, and then co-stain by immunofluorescence for macrophage markers. And so you can go to your tissue sections, and it's not live cell imaging like John Candelas would do or something, but you can say how many of these macrophages have epithelial staining inside, and they're calling it like a immunofluorescent phagocytosis assay. Because I've spent a lot of time trying to figure out how can we measure in samples we already have. And that's one way that Mehdi discovered on his own.
Mm-hmm. Actually, if I can just ask, since you said you are now studying breast cancer, do you think this actually change in macrophage behavior could have implications in the progression of breast cancer or tumor-associated inflammation?
Yes. Okay. So we have done lots of work on the role of macrophages in breast cancer and the normal breast, as well as the tumor-associated macrophages. And, you know, we thought, oh, these aren't using glucose. What's going to happen? Because actually the cancer cells communicate with the macrophages and vice versa. And so it's been shown that the cancer cells suck up lactate from macrophages. So we thought, what's going to happen? These are going to have smaller tumors. And those are ongoing studies right now. And so we have, you know, we have some pilot data using 2 different models of breast cancer. And hopefully we can finish that up with my postdoc now.
Well, that will
Hi,
be
Gökhan.
Thank you, Gökhan. That will be exciting study to read. I have one more question from Lee Kyang. Go ahead, Lee.
Hi, Liza. This is really enjoyable. So I have 0 background on macrophage, so just learn so much thing new. And simple question. So since you say that the macrophage seem to have this fuel preference, use glucose. My question is, under physiological conditions such as fasting and refeeding, is there any M1, M2 switch, at least in adipose tissue?
Okay. You know, I have not been 100% in that field. I'm lately going more to cancer meetings. I don't know the answer. It's probably done. I mean, you know, timed feeding, fasting, refeeding, these are all important. You know, I mean, there are papers probably from the early 2000s showing like if you increase glucose levels with streptozotocin in mouse models, or like you can change the macrophage phenotype. And I know I don't remember the first author, but I cited them in the paper. So but it's such a difference between a physiologic measure of glucose or whatever metabolite versus actually what's happening in the tissue. Because and this is what's cool about cancer growing, it's just like adipose tissue. So you start with something small, but it's expanding, and you could get hypoxia here, but not here. And so you could have these variations in lactate or whatever, even in the same tumor or even across an adipose tissue. And that's why it's so important, I think, to do cell biology and look at the cells and look at different regions of tissue. But those, I could email you. I could get the answer and email you.
Okay, thank you.
So single cell metabolomics would be great for those tissues, right?
Yeah.
Okay, one more question, and then we'll move on to Lee, actually. So Zhu, sorry.
Hello, I just figured out how to ask question. Yeah, actually I have two quick questions. So the first question is, you show that GLUT1 knockout macrophage have increased ROS. So I just wonder, have you checked the DNA damage? Because this pulse have the oxidative DNA damage. So why the GLUT1 deficiency going to affect cell cycle and lead to decreased proliferation? The second question is, you show the increased cholesterol in the plasma. So I just wonder, this increased lipid, the lipid. So have you checked the liver lipid metabolism to figure out, what is the cause of the increased plasma lipid?
Okay. That's a good question. These are good questions. So first of all, the knockout had less oxidative stress, less ROS. But in either case, more or less, we didn't look at any DNA damage. I didn't look at anything like that. I don't know anything about cell cycle. And, for most of my career, it was thought that macrophages are terminally differentiated until these incredible lineage tracing studies came out showing that, yolk sac derived macrophages are proliferative. It was really exciting in our journal club. And then your second question was about liver. I mean, it was just a matter of human beings to do the experiments, and we had liver. They're in the freezer or it's sectioned, and we just never did anything because it was too much for the paper, and I didn't have a liver expert, and we just couldn't pursue it. But macrophages are everywhere. That's what's so incredible about them. And we could talk more if you're interested.
Sure. Yeah. Thank you.
Thank you. I just wanted to comment that every time you say macrophages are everywhere, and Katie's eyes are gleaming. Okay, so there's a lot of macrophage lovers in this audience, as far as I know.
Yes.
Thank you, Liza, so much. And I'm going to hand it over to Lee and Lale.
Hey, thank you, Liza. It was a great talk. So our second speaker is Lee Chang, who's a great friend and a scientist in the Department of Pathology at Columbia University, New York. Lee's lab studies the pathophysiology of diabetes and obesity with a specific interest in the transcription factor PPARy gamma. And today he will talk about PPARy gamma deacetylation and its role in cardiometabolic diseases. Lee, it's all yours.
Thank you, Lali, and also thank you, Ever, for organizing this great seminar. This is a way to keep us active and alive in these pandemics. And so for now, right now, most of the time without the seminar, I have to, watch across the Hudson River to look at the Columbia campus where Lali and I work. So this is the George Washington Bridge here. This is where Columbia is. This is New Jersey. So today we'll talk about our newest discovery on the PPARy gamma and also some of its potential function in atherosclerosis. And also I'd love to hear your feedbacks and also comments on it. And also, by the way, so Lali and I know each other for a long time, and my lab mainly focus on adiposite. And because of Lali, and I can have stay close interaction with Dr. Eric Tabor's Alan Toss lab, and that help me to keep in this atherosclerosis loop. So thank you so much, Lali. And so I think for this audience, there's no need to mention how important our research is. So, and we know that so this cardiometabolic disease generally start with obesity, meaning too much fat accumulated in adiposite. that will cause insulin resistance in muscle, liver, that will then induce hyperinsulinemia, eventually beta cell burnout, have a result in the hyperglycemia stage, that is type 2 diabetes. Then this is like a climb on the mountains to take years to develop. However, once you reach this type 2 diabetes stage, then all these comorbidities start to develop and in a much faster way. And this like this go down the hill. And that this comorbidity increase atherosclerosis, stroke, heart disease, hypertension, dyslipidemia, and liver steatosis. That's all related to our cardiovascular health. So for this cardiometabolic disease, I think they have this kind of features. One is that they are epidemic. Second is they are chronic, take years to develop. Third is they involve multiple organs. So the challenge for us is how to improve metabolic dysfunctions in multiple organs simultaneously, not only in a single organ, but have to involve in multiple organs, such as fat, such as the liver, and also involved in macrophages and heart. So that require some factor has to be versatile and also metabolically important. And also this ideally this factor should be druggable. So for that, I think PPAR gamma is a good candidate. This is a hormone nuclear receptor, and it's really important for adipocyte biology. Indeed, this is the most important factor in adipocyte. And it requires ligand to bind. Once ligand bind, it hydrodimerize with RXR, bind to the DNA and start to regulate gene transcription. And another aspect of my lab focus on is protein post-translation modifications, the PTMs, and primarily acetylation. So for PPAR gamma, so PPAR gamma has so many functions. You can see from this nice review, it functions in so many organs, like in fat and in brain tissue, liver, pancreas, muscles that regulate metabolism, and also it also abundantly expressed in immune cells such as macrophages, and then to regulate inflammation. And unfortunately, activation of PPAR gamma will cause problem. Like in the heart, will cause the heart growth and also lipid storage that will cause some heart disease risk. And also in the kidney, it will cause the fluid retention that will also increase the risk for congestive heart failure. And in the bone, it will cause bone loss. And in the brain, it increases fluid intake. So PPAR gamma have many phases, and we need to better understand of its function. And then for the second aspect, so this is regarding the protein post-translation modifications, the PTMs. So we know that our genome contains about over 20,000 genes with alternative promoter and also alternative splicing, RNA editing. We end up with about 100,000 transcripts. With post-translational modifications, we can have over 1 million proteins. So these PTMs can sense environmental changes and also take place on existing proteins that need to synthesize a new protein. And they are also very important in regulating protein activity. For example, AKT requires phosphorylation to be activated. And also it regulates subcellular localization. In the case of FOXO1, phosphorylation will exclude it from nuclei and in cytoplasm. This is the way to inactivate FOXO1. It also regulates cofactor binding and also protein stability. Thus, this PTM regulation seems, at least in my understanding, is a very efficient and also economic way to regulate a protein's function. That with its large increase of the complexity of proteins, I think it perfectly underlines the complexity of metabolic disease. So years ago, when I was in my postdoc training with Dr. Mimo Achille, we discovered that the PPAR gamma have acetylation, and this cofactors CBP is the acetyltransferase. What is really intriguing to us is that we found that PPAR gamma ligand, this is a TZD, which is a potent insulin sensitizer, a full agonist of PPAR gamma, can induce PPAR gamma deacetylation on 2 lysines. It's not on the others, but there are multiple acetylation sites on PPAR gamma. But it only causes deacetylation on lysine 268 and 293 in the presence of 31, which you may know that is a protein deacetylase require NAD to activate. And later it regulates metabolism and also being proved as a longevity gene in yeast. So we further found the function of PPAR gamma deacetylation summarized here very briefly. So PPAR gamma, when PPAR gamma is acetylated, it will favor lipid storage in adipocyte that will contribute to insulin resistance. And in the presence of PPAR gamma full agonist, here is TZD, and also when 31 is active, it will recruit 31 to PPAR gamma and to cause the deacetylation on these 2 lysine residues. This deacetylated PPAR gamma will induce the brown remodeling of white adipocyte to increase energy expenditure and also insulin sensitivity. So this work has been published for a long time, and then we questioned what's the physiological function of PPAR gamma deacetylation. So we want to understand what's happening in vivo. So for this aim, we generate this deacetylation mimetic PPAR gamma knocking mice, which called 2KR. So that is to mutate the lysine into arginine. That we will keep the positive charge on the side chain. These 2 charge lysine also have the very similar length of side chains. Thus, this will maintain the positive charge, but abolish its acetylation potential. This is the way we generally do to mimic deacetylation. And because the PPAR gamma have so many functions in so many tissues, that's why we made this whole body knocking. Hopefully, we can capture the most of PPAR gamma's function in different tissues. So just a long story short, this work also being published 2 years ago. So we found a very interesting phenotype of this mouse, which we named the 2KR. So this so this have this dream couch potato phenotype, meaning that so unlike the wild type mice, the 2KR mice do not like to move. They sit there, keep eating. They eat about over 50% more food than the wild type, but they stay lean and also healthy. And because they have increased energy expenditure, and also because this is attributed to the brown remodeling of white adipose tissue, and because of this increased energy expenditure, these mice are protected from diet-induced obesity, particularly the visceral obesity, and also the obesity associated glucose intolerance, and also improve the liver steatosis. So we know that for the in diabetes patient, so there is increased risk for atherosclerosis. So the CVD is the leading cause of death in diabetes type 2, about 40% death. And the type 2 diabetes also increase the morbidity and also mortality of atherosclerosis about 4-fold. And very disappointingly, so no matter how hard we control glucose, it may improve the macrovascular health, but not the macrovascular health, meaning that not reduce the atherosclerotic risks. So this is remain a challenge for the field, how to reduce the atherosclerotic risk in type 2 diabetes. So we know that there is a large amount of study that showing that PPAR gamma activation actually is anti-atherosclerotic. It functions in macrophage, smooth muscle cells, and also endothelial cells. So we asked whether our deacetylation can also have some benefit to atherosclerosis. So to this end, we generate this 2KR mice on the LDL receptor knockout background. So this mice we put this mice on the Western type diet, as you all do. And very interestingly, this mice showed the reduced lesion at only this arch region here. You can see they reduce about 50% of the arch lesion, but not at the aortic root lesion. And this isoprotection is not due to metabolic improvements. These mice have the comparable body weight, similar glucose intolerance, and also similar insulin sensitivity. Neither is caused by any change of lipid profile. So they have a similar triglyceride levels, similar NIFA, and similar cholesterol, including HDL, VLDL, and LDL. So metabolic phenotype is very minimal in these mouse models by deacetylating PPAR gamma. So how to explain this isoprotection in this mice? So we did a very rough bone marrow transplantation in which we switched the bone marrow between wild type and the 2KR mice. And disappointingly, and this 2KR mice, even with receiving the wild type of bone marrow, they still have this isoprotection compared with the wild type mice receive the 2KR bone marrow, suggesting that macrophage unlikely account for this isoprotection by PPAR gamma deacetylation. And so we keep looking for the mechanism, and we found that indeed in the aorta, look at this isogeneric genes, ENOS, INOS, NOX2, and also this VCAM1, all is downregulated. This NOX4, which in some studies show is isoprotective, is increased. And also for this, the circulating IL-6 also decreased, suggesting that this might have decreased systemic inflammation. So this indeed happened in endothelial cells. So when we isolate the primary endothelial cells, we found a similar repression of INOS, NOX2, and IL-6 by the PPAR gamma deacetylation rather than this rosiglitazone. So then we questioned whether this deacetylation will improve vascular function. So with the help from Maria at NYIT, we found that PPAR gamma deacetylation increase the endothelium dependent relaxation as induced by this acetylcholine, but not to this endothelium independent relaxation induced by SMP. This decreases the minimal dose, effective dose of acetylcholine, and also increase the maximal relaxation. And there is no structure change of aorta between 2KR mice and the control mice. So here come to the clinical challenge. The biggest challenge in PPAR gamma research actually is the safety concern. The function or the importance of PPAR gamma is well established, but the safety still cannot be resolved. And the thing is, right now there is no better choice of insulin sensitizer than PPAR gamma agonist TZD. So we have to live with TZD, but it's not the ideal compound. And so we noticed that so in the 2KR mice, so by deacetylation, it doesn't change all the downstream target of PPAR gamma. It selectively activates some genes related to browning and lipids oxidation, and also repress some of the white adiposity-specific markers such as adenosine, without a strong impact on the regular PPAR gamma downstream target genes. So we asked, actually, this is not a question. So it's indeed our wish that can PPAR gamma deacetylation alleviate TZD side effect? And then we start to survey the TZD, the main side effects. So the cardiac risks, fluid retention, and bone loss. So we know that the TZD will increase heart hypertrophy, and like seen here with ROSI treatment, and the heart size is increased. but this increase is abolished in the 2KR mice. It can also increase the heart function in the 2KR mice, as shown here. I think you understand this result better than me. And in regard of fluid retention, which contributes to the congestive heart failure, so it will also cause the hemodilution of our blood cells. So that's why we measure the hematocrit as a parameter to assess fluid retention. We notice that in the wild-type mice here, after ROSI treatment within 2 weeks, it will cause this decrease of hematocrit in wild-type mice, and become very dramatic at 5 weeks treatment. And then in the 2KR mice, this is kind of delayed or inhibited. So the 2KR mice have the alleviated fluid retention. And then for bone loss is the most prevalent side effect of TZDs. And when we challenge the mice with ROSI for 5 weeks, we induce a significant bone loss in the wild-type mice, but this is completely prevented in the 2KR mice. And because in humans, we have to take the diabetes drugs for years. So then we try to extend our treatment time. This time we treat for 24 weeks to mimic the the people on the pill for about 10 to 20 years in humans. And we can see that this protection of bone by PPARy gamma deacetylation still there. And it increased the bone mineral density and also preserve bone volume. So how does PPARy gamma deacetylation improve skeleton health? So we know that in the bone, that adipocyte and osteoblast, which is the bone forming cells in the bone, actually share the same progenitor, that's the mesenchymal stem cells. So with the PPARy gamma activation, it will inhibit these stem cells to differentiate into osteoblast, but promote them to differentiate into adipocyte. That will cause the decrease of bone formation and eventually cause the bone loss. So then we question whether this also affects the bone marrow adiposity. And this phenotype actually you can tell by eye. You don't need to any instrument. So the bone marrow color in the wild-type mice is yellow after ROSI treatment because they have a lot of fat here. And in the 2KR mice on the TZD treatment, they still remain this red color, meaning they have a relatively healthy bone marrow. And this is the microscopy scanning of the lipid volume. This golden area is the lipid droplets here. You can see that it strikingly protects the bone marrow fat accumulation in the 2KR mice. And notice that in the bone marrow, that there is this lipid. These are the adipocyte. This is the bone marrow. So it's bone marrow and bone marrow adipocyte and also bone forming osteoblasts actually sit next to each other. So in the field we always know this, that with the bone loss, that there's always increase of bone marrow adiposity. People think that this bone marrow fat just as a cavity filler in the bone. When the bone loss, it has some cavity there, bone marrow and the adipocyte just go there to fill the space. And so we question whether maybe there is some paracrine regulation by adipokine that bone marrow adipocyte directly regulate bone remodeling. So we start to look for this potential adipokine. When we identify the adiposine as the repressive target of PPARy gamma deacetylation, you can appreciate that in the 2KR mice on high diet, the circulating adiposine is dramatically decreased. This can compare with adiponectin, another adipokine. And then on ROSI treatment, this is almost completely gone in the circulation. But this is still much stronger than adiponectin. So adiposine is indeed the first identified adipokine, and in 1980s, and also it is the most abundant protein produced by adipocyte. So we noticed that in 2 bone loss models, first is the TZD bone loss models. In the bone marrow, there is a huge induction of adiposine, and much more stronger than adiponectin. And we also employ the calorie restriction, which is another widely used bone loss models. We can also see this significant induction of adipose. That's why we speculate adipose maybe is involved in bone loss. As we get the adipose knockout mice from Jim Slow at Weill Cornell, and we first challenged the mice with TZD. We noticed that these mice, as we predicted, these mice have increased bone mineral density, both the cortical region and also trabecular region, with the lower bone marrow fat accumulation. And also in the calorie restriction, we see the only the increase of the protection of bone mineral density on this trabecular region, and also similar decrease of bone marrow fat there. So adiposine, besides it's an adipokine, it's also being known as the factor D in the alternative complement pathway. This activation of this factor D eventually lead to the activation of C3. Thus, C3 is a downstream mediator of adiposine. So then we get the C3 knockout mice to examine their response to TZD and calorie restriction. So very similar to adiposine knockout mice, this C3 knockout mice have a better bone mineral density and also much less bone marrow fat compared with the wild type mice. And also in calorie restriction, this remain the higher bone mineral density and also lower bone marrow fat, bone marrow adiposity. So that indicated that adiposine mediate the bone protective effect of PPARy gamma deacetylation. Then the following question is, how about this protective effect? So for this, we crossed the adiposine knockout mice to LDL receptor knockout mice. And very disappointingly, these mice have no phenotype. Although adiposine is so abundantly produced, the level in the circulation is so high, and it's closely related to inflammation. And they just have nothing to do with atherosclerosis at both the root region and also at the arch region. So blunted phenotype. So to put this together, so I think PPARy gamma deacetylation can uncouple TZD's cardiometabolic benefit from adverse side effect. It functions to increase fat remodeling and also increase energy balance, and it also reduces the cardiovascular risks by this industrial protective effect. And in the bone, it promotes bone formation, decrease bone, inhibit bone marrow fat development as this functions through adiposine. And so overall, it alleviates TZD side effect. That's making this a promising target to develop the safer insulin sensitizer. So this is my understanding of the PTM on PPARy gamma. So we think that the PTM is really critical for the transcriptional selectivity of PPARy gamma. So at the basal level, PPARy gamma activity is really low. Then when we give them the full agonist TZD, it will cause the full activation of PPARy gamma in all the tissues. This causes the trouble for the side effect of TZDs. But under the physiological conditions, I think the PPARy gamma is not fully active. In some cells, it needs to be repressed. Some cells need to be activated. And some cells need to higher activity, some need lower activity. So this is the I call we need the transcriptional selectivity of PPARy gamma rather than its full activation. This can be achieved from the data I showed you through protein deacetylation. And there have been some study in the field that are trying to develop some partial agonist of PPARy gamma to achieve this goal. So far without any success. The best result, to my knowledge, is that they have this partial agonist treated the mice for only 4 days, can alleviate fluid irritation. That's the best result so far we know for the partial agonist. So I think that PTM is so far seems that it's more promising than the partial agonist, and this it functions on the molecular level through the recruiter cofactors and also specific downstream regulate specific downstream targets. So so this one we right now there is some work ongoing in the lab. So we are very lucky that we collaborate with a chemical lab. We found a chemical that can specifically target the PPAR gamma acetylation that can associate the side effect from the benefit of PPAR gamma. So that's something we are right now we are working on that. Hopefully next time I can share with you these exciting findings. So with that, I'd like to thank my lab people. This ISO study mainly done by my former postdoc, Dr. Longhua Liu, and also Dr. Li Hongfan. And the bone part been done by Nikki and also the other lab people in the lab. So I also like to appreciate my collaborator and friends at Columbia, my mentor, Dr. Achille, my colleagues, Dr. Pajivani, Becky Heusler, and Lali. And also thank Dr. Tibers and Tao, and also Dr. Guo helped us with the bone analysis. I'd like to thank my collaborator, Dr. Maria Carrillo Sepulveda at NYIT, helped with us with the endothelial cell study, and also Dr. James Lowe at the Weill Cornell for providing us the adiposity, so, and also our founding results. I think, am I right on time, Lali?
Yes, thank you.
Great.
All right, great. Thank you, Lee. It was a great talk. I know people have questions, but if you don't know how this is going on, if you have any questions, please use the raise your hand function at the bottom of your screen. I'll start with the first question, Lee. So you mentioned about the bone loss phenotype of TZD, right? Within the 2KR mice, that it's through adiposity, for example, right?
Mm-hmm.
But do you know what the mechanism, for example, of this TZD side effects or KR's protection in terms of the heart size, or like fluid retention and heart size? Do you have any insights on that, the mechanism?
Okay, so for the heart, so I really not familiar with the heart, but we only briefly survey some of the PPAR gamma downstream target genes identified by Dr. Eric Goldberg. So he overexpressed PPAR gamma in the heart, get this cardiac toxicity of TZD phenotype. So we found that the deacetylation of PPAR gamma actually inhibit all these lipid storing genes in the heart. I think, and also increase the lipid burning genes expression. That's my minimal knowledge on the heart cardiac beneficial of PPAR gamma deacetylation.
Okay. All right, great. We have many questions. Let's start with Mari Guaretti. Mari, go for it.
Lee, wonderful talk. I really enjoyed it. I was wondering if you know if PPAR gamma has a role in vascular calcification, and if you think then that PPAR gamma deacetylation might promote vascular calcification as a negative consequence.
And Mary, thank you so much for this great question. And again, so I don't know much about this vascular calcification, but there is the best phenotype of this model is in the vascular system, so, and also in the bone. And we know that there is improved vascular function, and also, and my collaborator, Dr. Maria at NYIT, also she's working on this. I provided her the mice. She showed some really compelling data that these 2KR mice are protected from obesity-induced hypertension. So I guess that there's something related to that, but I don't know how exactly it functions.
Okay, and next question is Juwei Zhu.
Yeah, okay, Xuewei.
Yes, go ahead.
Hello, I have a quick question about your 2KR, the bone phenotype. I don't know whether I missed or not. So my question is because we know PPAR gamma also expressed in macrophage in the osteoclast. So I just wonder, in addition to osteoblast, have you seen maybe this 2KR actually affect osteoclast differentiation and function?
Yeah, so we did check the effect of osteoclast differentiation and activity. There is not much change on that. Okay. So the effect is mainly on the osteoblast side rather than osteoclast.
I see. Thanks.
Okay, and then next question is from Dawn Fernandez.
Hi, what a wonderful talk. It really was. I really just had a question regarding the deacetylated form of the protein. Did you ever do any proteomics like on these wild type versus deacetylated forms? And also, why is it that you end up having different transcriptional regulation patterns?
Hi, Lars. This is a great question. And this is something we're trying to focus on now. And we are trying to compare their cofactors of PPAR gamma. I guess that this modification will modulate the cofactors. For example, this deacetylated form will preferably bind with the PRDM16, which is the Browning regulator. And will also decrease the interaction with the NCOL, a co-repressor. And the other potential mechanism is through modulating DNA binding. So this actually is also surprising to me because this acetylation site is on the ligand binding domain on PPAR gamma. At the beginning, I didn't think it will change the DNA binding of PPAR gamma. Then we did some chip. We found that on the adiposity promoter, indeed, it decreased PPAR gamma binding to the promoter. And then when I looked at the literature, I found that this acetylation site actually is seated in the between the ligand binding domain and also DNA binding domain of PPAR gamma. Thus, it may directly regulate the DNA binding. So we are doing some chip on this, chip-seq on this now.
Did you happen to ever check any of the phosphorylation effects as well? So I mean, maybe it's a combination of the post-translational modifications why you might see differential effects.
So you must be an expert in this field. And yes, we did check phosphorylation. And we know that PPAR gamma acetylation go along with acetylation. So when there is the acetylation, there is always increased the PPAR gamma phosphorylation. When it's deacetylated, phosphorylation of PPAR gamma also decreased. However, recently I noticed a study that they use the PPAR gamma phosphorylation deficient PPAR gamma knocking mice. And this might have a minimal phenotype there. So that's why I don't think it functions through phosphorylation.
It was really lovely. Thank you.
Okay, thank you so much.
And we have last question from Mabruka Al-Faidi.
Hi, that's really very interesting talk. I'm also interested in the deacetylated form and the phenotype that you saw in mice. So it's very interesting to see that it's a very local area of the aorta that shows changes in atherosclerosis. So I was wondering, since you saw changes in gene profile in that aorta, is it local changes in the gene profile, or when you assess, did you assess the whole aorta as a whole? And I was wondering, why did you see reduction in PCAM, for example, and pro-inflammatory mediators, and there is an increase in NOX4? Thank you.
Okay, thank you so much, Mabruka. So yes, this question I'm also intrigued in, and then to be honest, once it ends up in the endothelial cell, it out of my comfortable zone. I don't know how to deal with the endothelial cell, and I have to have a sudden stop there. And I love to share this mouse model, whatever material we have with some endothelial cell expert to pursue this answer. I also really want to know this answer.
That's very interesting. Thank you.
Thank you.
Okay, I guess that's the end of the questions. Thank you again, Lee. It was a great talk. I guess we're concluding this session today. We would like to thank Liza and Lee again for wonderful presentations, and also the questions. Those were all really enriching. So we'll see you next week with 2 different speakers.
Okay. Thank you all. Thank you, Ivor and Lali.
Bye.
Bye. Nice to see you.
Thank you guys. Thank you.
Bye.
Bye.
Happy weekend.
Have a good weekend.
Nice weekend.
Bye.